Now you can hear me and see me?
Yes, very clearly.
Uh, uh, exactly. So thank you very much for inviting me. Today I will talk about the antimicrobial peptides against multidrug-resistant pseudomonas aeruginosa from biofilm from cystic fibrosis patients. So let me shock you. Superbugs are on a race. There is around 300 estimated 300,000 deaths because of antimicrobial resistant pathogens. And by the World Organization, we're going to reach around in 2050 around 10 million deaths. So it's not only the economical, it's not only the death is the issue, it's also the economical issue. It's going to cost us a lot. And just to let you a reference, it's around 6.76 death out of COVID-19. And we are in discovery void. In the last 30 years, there is no new antimicrobial agent that got into the market. So this is why we're working on antimicrobial peptides. Antimicrobial peptides are potential therapeutics. They are not, as you can see it. It's can be in bacteria, also in trees, other species. They are not conserved by their sequence or secondary structure. So what does make them antimicrobial peptides? So they are part of our immune systems from all life forms, and they share this stuff. They are short, up to 40 amino acids. The overcharge is between 2 to 9. So it means that they're positive and they are hydrophobic. This characteristic These properties allow them to interact with the membranes. Now, antimicrobial peptides you can divide two classes. One of them is to modulate the immune system, and the other one to kill microbes. And now we will focus on killing microbes. These negatively and positively charged amino acids allow them to bind to gram-negative bacteria. It could be either any pathogen, it can be positively charged, and even cancer cells, viruses, fungus, a lot of them. But in this case, I will work on gram-negative cells. So those amino acids, electrostatic interact with the membrane because of the positively charged. The LPS is negatively charged. In other membranes, it's other stuff that are negatively charged. And the hydrophobic parts, the hydrophobic amino acids, allow them to interact, to penetrate to the membrane. They're working, all of them, bothly a lot of, there are more the same. There are a lot of models. There's the barrel state model, the toroidal models, and the carpet model that actually my supervisor Yechiel Shai was the one that discovered it. They all work the same. They are interact with the membrane, the pathogen membrane, and by the gradenic-like manner, they are doing pores and metallization, and all of them leading to cell death. Our bacteria, in this case, we worked on the Pseudomonas aeruginosa, that is opportunistic, it's gram-negative bacteria. It's pathogenic to immunocompromised patients. It can be found in any surface, and this is why it's a huge problem in hospitals, that it's really hard to get rid of it. And one of the features he has is can form biofilm. Biofilm are microcolonies that work together. They start with planktonic cells that attach to any surface and then form a microcolonies. These microcolonies extract polymetrics that gives in structural stability and gives resistance by that to antimicrobial agents like antibiotics. And this is why it's more resistant to antibiotics than other stuff. The whole process is a cycle because it goes again when the planktonic cells detach from the biofilm and attach again to a new surface. In our lab, we're designing peptides, as you can see in the first one, then we chemical synthesize, then we purify, check their physical and chemical evaluation, and check the antimicrobial properties of it. Now, why to use antimicrobial peptides that are synthetic? Because the natural antimicrobial peptides, as I talked about them, they also toxic to eukaryotic cells. we started with a synthetic antimicrobial peptide that is form of L-amino acids, K6L9, 6 lysines, and 9 leucines. As you can see in red, there is the all positively charged amino acids, and all the hydrophobic amino acids, it's the L ones. We segregated, but this peptide is amphipathic. What it means is amphipathic? When it forms its alpha helix, it becomes all the lysines are in one side and all the leucines are in the other side. Now we took this peptide and we replaced some amino acid with the amino acids with it. Then we segregate these peptides, and there is the seg5L, as you can see, well, the all leucines in the center and all the lysines in the side, where seg5D has the same sequence as seg5L, whereas there is the same amino acids replacement in the same positions. The opposite we did with the seg6D and seg6L, and we took the well-known antimicrobial peptides LL37 as a reference. As you can see, this is for summarize, the secondary structure is disrupted, and over here is also there is a charge distribution alternation to check what is the charge distribution contribute to the process. The length is the same length, and then the charge and the molecular weight is also the same. The hemolysis, as you can see, the incorporation of the amino acids to the peptides eliminate the toxicity effect, the hemolysis effect. Now, the MIG, actually with the amino acids, it's actually better. As you can see, the F1D is more potent than the F1L. The same phenomenon occurs in seg5L compared to the seg5D and the seg6L and the seg5D, where you can see that the amino acids are more potent than the L form of the same peptide. This was also confirmed by the bacterial killing. Now we check the inhibition effect of this peptide, and as you can see, the incorporation of the amino acids inside the sequence is better. Here, the same phenomenon occurs also in seg5D and seg5L. Compared with the amino acids, is better than the L form. As you can see in y-axis, the biofilm biomass, and we checked the MIC concentration is the minimal inhibitory concentration, and we got lower and lower. So the amino acids partial replacements improves the biofilm inhibition activity. But what happened with the degradation activity? So when we actually put the biofilm and it's more potent, so as you can see, the diameter partial replacements improves the biofilm degradation activity. Then we also check is the anti-biofilm that depends on the secondary structure. So actually, we checked with the CD and FTIR, and it's not. The F1L is alpha helix, and all the others have structure that is other or beta sheets. Then we wanted to see which is. Then we checked against multidrug-resistant pathogens from cystic fibrosis patients. So CF is a mainly respiratory system, and this is what happens where there is accumulation of sputum, and we can see that pathogens, mostly pseudomonas aeruginosa, like to accumulate over there. Then we check their antimicrobial, the resistant. As you can see, the red is sensitive and the gray is resistant, as most of them are resistant to antimicrobial. Then we check what happens about the activity of our antimicrobial peptides. So we checked seg5D, seg6D, and F1D, and the F1D is the most active peptide among all the isolated from the Pseudomonas aeruginosa from the patients that are resistant to antibiotics. So can they inhibit also? So we check that, and also the F1D is the most potent one among all the peptides. And there you can see there is a correlation between the concentration to the anti-biofilm activity, the anti-biofilm. Next, we wanted to see if they can degrade it. So also here, the F1D is the most potent peptide comparison to the other. And also over here, you can see there is a correlation between the concentration to the biofilm biomass. So our three peptide degrade pseudomonas are for the patient's biofilms. Next, we wanted to see if they can degrade in artificial sputum. So actually, only F1D stayed active in artificial sputum. Then we wanted to check what happens inside the sputum itself. So we put our peptide and we check three things. One of them is the stability of the peptide. Secondly is the efficiency inside against planktonic in the sputum itself. And next thing, what happens to the biofilm itself in the CF sputum. So if it's stable in resistant proteolytic, so as you can see, the L amino acid, the F1L, that is also like F1D, degraded after half an hour, F1L eliminated after 12 hours, and all the amino acids, peptides with the amino acids stay stable. So, and as you can see over here, they are also active inside the sputum itself. F1D stays stable. Then we wanted to see if it can degrade biofilm inhibition in patients' sputum. So we checked it by confocal, and the red is dead and the green is alive. And as you can see, the F1D, SEC5D are really potent, and the green ones where L37 lost its activity inside the sputum. And then we checked if there is a resistance. So we took our peptide for 45 days and we checked that. And as you can see, the resistance after a prolonged process, the gentamicin, the MIC folds raise up, where the antimicrobial peptides do not raise up. It means that it's really hard. It's also inducible and constitutive. It means it's very hard to induce resistance. The peptides do not induce resistance to the pathogen, and it's very hard to gain resistance against these antimicrobial peptides. So for summarize and take home message, synthetic antimicrobials have antimicrobial and antibiofilm activity in CF sputum environment. There are synthetic antimicrobials stable and resistant to proteases. Synthetic antimicrobials do not induce resistance, and there are promising candidates against multidrug-resistant pathogens. I would say thank you to my lab, Yechiel Shai Lab, and to our collaborators from Tel HaShomer Hospital. Thank you very much.
Thank you, Daniel. This is very important research, and I hope one day it will move on to a clinical trials phase, more applicable. We have a question from Payam and then another one in the chat. Payam can go first, and then I can ask the one in the chat.
Go on.
Daniel, can you hear me okay?
Yes, Payam.
Conference room, I'm not exactly sure how well we're connected.
Actually, I can't hear you, sorry.
Oh. Really wonderful talk. So I was curious in the case of cystic fibrosis, if having some sort of a specific immune suppressant that can actually enable the drugs to work better would be helpful to addressing also the drug resistance. Does that kind of make sense? Because I know that actually immune tolerance is also hampered in this system. There's a little bit of overt, non-specific immune activation that also contributes to pathology, right?
Yes. So this is the other part of our project. You just let's say you just raise it up. We know that our antimicrobial peptides have another system that can modulate the immune system, and there is another subject about the cystic fibrosis where the immune cells do not work well as they should be, and they are not clear the pathogens. So we know that our antimicrobials not only have the possibility or the ability to kill the pathogen, they also can neutralize the LPS recognition. So sometimes especially in cystic fibrosis patients, the problem is that the immune system inflammate all the area, and there is going to do lung failure. So we know this. This is our next investigation that we know that we search for. That we know that they inhibit IL-8, and this is how they can prevent recruitment of neutrophils.
Thank you so much. Great talk.
Thank you.
So Daniel, I'll just jump in with my question that was in the chat. So you mention in you show in your data always a % reduction in biomass, and I'm just wondering whether you've looked at different, biomass loads and whether the outcomes vary at all with the amount of biomass. Just a question.
We started to check that, but and the differences between them. The difference between the biofilm biomass and how has the peptides effect on the biofilm depends on the secretion of toxins by the biofilm. And we saw when there is a the high levels of toxins, this is where the antimicrobial peptides do not active. And this is one of the reasons we thought so, that those toxins has negatively charged, and this is why the peptides interact with them. So I would say that maybe it's a mechanism of the biofilm to protect itself. The problem is, or I would say so differently, it's not the biofilm problem is that takes a lot of energy from the biofilm to do so. So it will be very hard to maintain this ability.
Got it. Thank you very much.
Thank you once again, Daniel.